Welcome to the deep dive. Today, we're getting our feet wet, quite literally perhaps, thinking about wildlife conservation. We're diving into a 2024 paper. It's called Comparing the Effectiveness of Environmental DNA in Camera Traps for Surveying American Mink, Neogale vison. The lead author is Eleanor L.D. Girolamo.
Right. And our mission today really is to give you a shortcut. We want to unpack what this study found about the best ways to survey tricky species like the American mink, especially looking at that balance between, well, getting good data and what it costs in time and money.
And the American mink is kind of the perfect test case for this, isn't it? It's what we call a cryptic species.
Exactly, meaning it's elusive, hard to spot, avoids humans, often nocturnal.
Yeah, just generally a pain to study reliably. It's a secretive, semi-aquatic musculid. So getting solid, unbiased population data. Traditionally, that's been super time-consuming and, frankly, really expensive. We badly need ways to track them that aren't just based on, say, fur harvest numbers.
And knowing their status is critical. The mink isn't just hard to study. Ecologically, it's a real powerhouse. It's a mesopredator.
So, middle of the food chain.
Pretty much.
Yeah.
And it really shapes the local community. It's an intense predator on small vertebrates, think small fish like salmonids, amphibians, that sort of thing. If mink numbers change drastically up or down, the whole system feels it.
Right. And historically, we did track them, partly through economics, through the fur trade. Its pelt was desirable. I looked at some Indiana data. Between 2007 and 2017, licensed buyers there bought about 1,500 pelts a year on average.
But, and this is a big but, those numbers have been dropping pretty steadily since around 2012. Now, does that mean the mink population in Indiana is actually declining? Maybe. But harvest data like that is inherently biased. It only reflects what trappers managed to catch, not the whole picture.
It doesn't tell you about effort or market prices or anything else.
Exactly. So for real conservation status, you need independent monitoring. And there's another layer here globally, the American mink, where it's not native like in parts of Europe. It's a serious invasive species.
Oh, right. It competes with the native European mink.
Severely. It outcompetes the European mink, Mustela lutriola, which is critically endangered. They fill the same ecological niche. They look almost identical.
Wow. Okay, so telling them apart and tracking where the American mink is spreading is pretty vital for conservation over there, too.
Absolutely. Which brings us right back to this study's core question. How do the tracking methods stack up? You've got the traditional method versus the newer tech.
So the traditional one is the camera trap, or CT.
Yep. We know the pros. They don't disturb the animals much. They're fairly low maintenance once set up, relatively cheap to deploy initially. But they have downsides, especially for something small and sneaky like a mink.
Detection rates can be low, right? And if they trigger it at night,
the pictures can be pretty poor quality. You might know something was there, but maybe not definitively what or what it was doing.
Okay, so that's the old guard. What's the new contender?
Environmental DNA, eDNA. This is cool because it's also non-invasive, but you don't need to see the animal at all. You're looking for the tiny traces of DNA it leaves behind in the environment.
Like skin cell, saliva.
Skin cells, feces, saliva, mucus, all that stuff contains DNA, and it gets shed into the water. You collect a water sample, filter it, and look for that specific mink DNA signature.
And the advantage there is timing.
Yes, a key advantage is the recency. If you detect mink eDNA in a water sample, you know a mink was likely there relatively recently. Studies suggest the DNA signal degrades significantly after, maybe about 7 days or so in water.
So it's a snapshot of current or very recent presence.
Yeah.
Not something that lingered for months.
Exactly. Okay, so the researchers set up this head-to-head comparison. They did it during the mink's late breeding season, March to May 2021. 7 different sites in northeastern Indiana, ponds, wetlands, a creek. Different habitats.
And they picked those sites carefully, I assume.
Oh, yeah, very strategically. They made sure the sites were spread out, at least 3.4 kilometers apart.
Huh. Why that specific distance?
It's based on the average home range size for a male mink. They wanted to minimize the chance they were just repeatedly detecting the same individual animal across multiple sites.
Ah, smart. Avoid pseudo-replication, basically. Keep the samples independent.
Precisely. It avoids skewing the data towards one particularly active, wide-ranging male.
Okay, let's talk nuts and bolts. How do they actually do the surveys? The camera traps first.
So for the CTs, they ran them for a total of 504 trap days. That's across all sites. Typically 3 to 5 cameras per site, passive infrared ones, placed low about half a meter from the shoreline, maybe a meter up.
And importantly, no bait. Right.
Crucially, no bait or lures. They wanted the detection to be passive, just like with eDNA, to make the comparison as fair as possible.
Makes sense. Okay, what about the eDNA side?
They collected a total of 210 water samples across all the sites and visits. Back in the lab, the process involves filtering the water to capture the DNA, then extracting that DNA.
And then the fancy part?
Then the qPCR, quantitative real-time PCR. Think of it like a highly specific DNA photocopier that also counts the copies in real time. They used a newly developed probe, an assay designed specifically for American mink DNA.
Targeting a particular gene.
Yes, the cytochrome B gene, or CT. They tested it to make sure it wouldn't accidentally pick up DNA from other local mustelids like weasels or otters.
Okay, diligence done. Fieldwork finished. Lab work complete. Lots of effort. So, what did they find? Who won? Camera traps or eDNA?
Well, this is where it gets interesting. Individually, it was surprisingly close, almost a draw, statistically speaking.
Really? Tell me the numbers.
Okay, camera traps detected mink at 4 out of the 7 sites. That's 57% of sites. And the detection probability, that's row O, was calculated at 0.36.
Okay, 57% of sites, 0.36 probability. And eDNA?
eDNA detected mink at more sites 5 out of 7, which is 71%. But its detection probability was slightly lower at 0.25.
Hang on. That sounds weird. eDNA found them at more locations, but had a lower detection probability. How does that work?
Yeah, it seems counterintuitive at first glance. But detection probability isn't just about presence-absence across sites. It factors in the effort like the number of samples or trap days, and the chance of getting a positive hit on any single attempt or sample, assuming the animal is there.
Oh, okay. So more sites overall, but maybe fewer positive hits within those sites on a per-sample basis for eDNA.
Something like that. The statistical models they ran basically said there was no significant difference in the detection power between the two methods when used alone. The model that fit best assumed detection probability was constant for both. Essentially, if a mink's present, your chance of catching it with one camera check or one water sample is roughly similar for either method.
Okay, so individually, they're comparable performers, despite the slight differences in site hits. But what about using them together? That's often the key in these comparisons, isn't it?
Absolutely, and that's where the real power emerged, when they combined the data from both camera traps and eDNA.
Let me guess, detection went up.
Significantly. The combined approach detected mink at 6 out of the 7 sites. That's an 86% success rate in finding where mink were present, and the combined detection probability jumped to 0.47.
Wow. Okay, so 86% of sites confirmed with mink presence. That's a big improvement over either method alone. If you really want to know if mink are in an area, using both is clearly the best bet.
Definitely. For the most comprehensive picture, the combination is strongest.
But, and here comes the big but, for anyone managing a budget. We need to talk about the resources, the time, and the money. This was maybe the biggest reveal, right?
This was the real aha moment, I think. Let's start with time.
Which one was faster?
eDNA, by a decent margin. The whole eDNA process, including field work and lab work, took about 81.8 hours. The camera trapping, including setup, maintenance, and crucially photo analysis, took 117.5 hours.
So eDNA was nearly 30% faster overall.
Yep. And the main reason, you guessed it.
Processing the photos.
Exactly. Sifting through potentially hundreds of thousands of camera trap images. Most of them are false triggers, wind-moving branches, other animals, you name it. It's incredibly time-consuming, tedious work.
I can only imagine. Clicking through endless photos of raccoons and squirrels. Okay, so eDNA saves a lot of labor time. But what about the actual cost?
Ah, well, that's the flip side, and it's a stark difference. The eDNA survey cost roughly $9,263 USD.
Okay, and the camera traps?
The camera trap survey cost came in at about $4,320 USD.
Whoa, so eDNA was more than twice as expensive.
Yes, over double the cost. And the vast majority of that eDNA expense wasn't the field work or even the fancy qPCR machine time, it was the consumables.
Meaning?
The single-use lab supplies, filters, extraction kits, qPCR regens, all the specialized stuff you use up for each sample, that adds up very quickly.
Okay, so that's the core trade-off laid bare. eDNA saves you significant time, nearly 30%, but it costs you double the money, mostly due to lab supplies. That's a tough calculation for wildlife managers.
It really is. It forces you to decide which resource, time, or money is more constrained for your project.
Let's dig into the nuances a bit more. Did each method find mink where the other one missed? Were there unique detections?
Yes, there were. eDNA got positive hits at 2 sites where the camera trap saw absolutely nothing.
Okay, so eDNA found mink. The cameras missed twice. What about the other way around?
The cameras only got one unique detection. They found mink at one site where the eDNA samples all came back negative.
Any idea why the cameras might have failed at those two sites?
Well, one potential factor, especially at a public site like the Fort Wayne Children's Zoo, which was one of the study locations, was access. They couldn't put cameras along the entire shoreline because of the risk of theft or vandalism in high-traffic areas.
Right, so they had unavoidable gaps in their camera coverage there. eDNA sampling might have picked up signals from those uncamera-trapped zones.
Possibly. That highlights a practical limitation of cameras in some settings.
But what about the flip side? What do cameras give you that eDNA just can't?
Behavior. That's the huge advantage of camera traps. eDNA tells you if the mink was there. A camera trap, if you get a good image or video, can tell you what it was doing.
And they got some of that data here.
They did. They found most detections, 64%, were at night. And they saw mink doing different things. About half the detections involved swimming, others showed them moving along logs or woody debris near the shore, and some just sitting.
That kind of information, how the animal uses the habitat, is gold for conservation planning, right?
Mm-hmm.
eDNA doesn't give you that context.
Not at all. eDNA is presence, absence, maybe relative abundance if you get sophisticated, but not behavior.
Okay, let's talk about the eDNA limitations specifically in this study. You mentioned it detected mink at more sites, but the overall positive rate across all 210 water samples was actually quite low, wasn't it? Only 6%.
Yes, only 13 samples out of 210 came back positive. That's a low hit rate.
Why so low, do they think?
Several potential reasons. First, the mink itself. It's highly mobile, covers a large territory, and it's only semi-aquatic. It's not living in the water constantly, so its DNA might be quite diluted or patchily distributed in a pond or creek.
Makes sense. What else?
Environmental factors definitely play a role. They noted that at the creek site, detection seemed lower after heavy rainfall. The increased water flow could dilute the DNA signal or just wash it downstream much faster.
So the environment isn't static. Water conditions affect eDNA success.
For sure. And then there's the lab side. While they confirmed their assay was specific to American mink and didn't cross-react with other local mustelids.
Which is good.
Very good. But the sensitivity of their assay, basically how faint a DNA signal it could reliably detect, might have been a bit lower, or rather the detection threshold was higher compared to some other eDNA studies on other species.
Meaning it might have missed some very weak, low-level positive signals.
It's possible. There might be room to optimize the lab protocol further to potentially pick up even fainter traces of mink DNA in the future. Improve that sensitivity.
Okay, so summing up the findings. Combining methods gives the best overall detection success. Individually, they're statistically similar, but eDNA is faster but much pricier, while CT is slower but cheaper and gives behavioral data. So, what's the practical advice for a wildlife manager based on this?
Given the resource constraints most managers face, the paper suggests a kind of two-step strategy, playing to the strengths of each method.
What's step one?
Use eDNA first for broad-scale initial surveys. Because it's faster, you can cover more ground or more sites relatively quickly to just find out where mink are potentially present. It's efficient for that initial presence-absence mapping.
Okay, so eDNA tells you which ponds or creeks to focus on. Then what?
Then, step two. You go back to those specific sites identified by eDNA and deploy your camera traps there. Now you're using the cheaper, slower method more strategically, only where you have a high likelihood of the animal being present to get that richer data, the behavioral insights, activity patterns, maybe even estimate numbers if you get enough detections.
That makes a lot of sense. Use the fast, expensive tool for broad screening and the slower, cheaper tool for targeted, detailed follow-up.
Exactly. It optimizes the trade-off.
And if we want to make eDNA even more useful, especially as that first step, the key is bringing down that cost, right? Tackling the consumables issue.
Absolutely. Future research really needs to focus on refining the eDNA protocols to make them cheaper. One obvious target is sample volume.
How so?
Well, maybe you don't need to collect and filter, say, a whole liter of water per sample. If research shows that a much smaller volume, maybe just 50 milliliters, as the paper suggests, is still effective for detecting mink.
Then you drastically cut down on the cost of filters, extraction kits, everything per sample.
Precisely. Finding that minimum effective sample volume could make large-scale eDNA surveys much more economically feasible. And improving assay sensitivity would also help boost detection rates without necessarily increasing cost per sample.
Okay, let's loop back quickly to that European situation, the invasive American mink versus the native European mink. How does this study inform that conflict?
Well, it reinforces that eDNA has huge potential there. Because the two species look identical, you can't easily tell them apart with cameras or visual surveys. eDNA could be the key non-invasive tool to map where the American mink is spreading and displacing the European one.
If you can tell their DNA apart.
That's the crucial next step. You need an assay, a DNA probe that is hyperspecific and can reliably distinguish between Neogale vison DNA and Mustela lutriola DNA, even when they're mixed in the same water sample. Developing and validating such an assay is critical for using eDNA effectively in that European conservation crisis.
Right, so this deep dive really clarifies the current state of play. For elusive species like mink, eDNA is this powerful time-saving tool, especially for initial surveys or in tricky locations. But it comes with that hefty price tag for consumables right now.
While camera traps are cheaper overall and give you that invaluable behavioral context, but they take a lot more labor time, especially in post-processing, and can miss things.
Then the absolute best detection comes from using them together, ideally in that strategic two-step approach.
That seems to be the most efficient path forward currently.
Okay, let's wrap up with a final thought for our listeners. We've seen this clear trade-off. eDNA speed versus its current cost, driven by lab supplies.
But technology evolves, right? Costs for things like DNA sequencing and reagents have generally trended downwards over time as techniques become more widespread and refined. Assay sensitivity improves too.
So here's something to ponder. As those eDNA lab costs inevitably decrease and the methods get even more sensitive, will that sheer time efficiency being nearly 30% faster eventually make eDNA the absolute go-to first step for surveying most difficult-to-track predators like mink almost everywhere?
Even if it means initially giving up that immediate behavioral data you get from cameras, will the speed and scalability of eDNA become so advantageous that it outweighs the initial lack of behavioral context for broad-scale monitoring?
That changing balance between cost, speed, and data richness. That's definitely something to keep an eye on in wildlife monitoring.